Hi, welcome to week 12 of the course. And this week we are starting to talk about other omics besides just general genomics, whole genome sequencing. This time we're talking about sequencing methods related to epigenetics. Epigenetics refers to modifications to DNA or chromatin structure that aren't necessarily changes to the A's and T's and G's and C's of the DNA sequence. This is an image that was in a previous video from the semester where we showed that there are levels of compaction that make up the mitotic chromosome. So if you go from the naked DNA, which is mostly what we've been talking about for the last couple of weeks now, that naked DNA in your cells right now is actually packaged on histone proteins, here shown in kind of gold, and those histones are compacted in various configurations that make up compact chromatin that make up your chromosomes. I want to cover two elements of epigenetics today. The first is histone modifications. These are reversible post-translational modifications that occur to modify the chromatin structure. They're reversible, they can be added or removed by enzymes in the cell, and they're post-translational, meaning this is a modification that happens to the histone protein after it has been translated. And then I want to talk about DNA methylation. This is, again, a reversible modification that occurs directly to the A's and T's and G's and C's of the DNA sequence. In this case, it is just happening to the C's, to the cytosine residues, but this is not a modification to a protein, it's a modification to a base of DNA itself. Both of these types of modifications come in many different flavors. What I want you to really take away from this is that these modifications can make DNA more or less compact. So that changes what types of cellular machinery can access the DNA. So first, talking about histone modifications, one type of histone modification is histone acetylation. I'm going to use this diagram over the next couple of slides, so let me take a second here to orient you to it. So this is what we've already seen on a previous slide here. So we have our compact mitotic chromosome, and we're kind of unraveling it to reveal this beads on a string configuration. And what's also being shown here is that these beads, these are nucleosomes, which are made up of different histone proteins. Um, those histone proteins have tails. So specifically, this is looking at the tail of histone H2A. That's one of the components of the nucleosome, the bead. This is the tail of histone 3. This is the tail of histone H2B. And this is the tail of H4. This is just a diagram shown to illustrate some of the types of modifications that can happen. So first we'll look at histone acetylation, which you can see here in purple on the diagram. So you see that some acetylation is happening on this lysine 5 on histone H2A, and on, so we have here another lysine, a lot of lysines in histone 3 and histone 4 and H2B. If you can see, these acetylations are often, always occurring on lysine residues on these histone tails. Another type of histone modification is histone methylation. So instead of adding an acetyl group to the amino acid residue, this is adding a methyl group. So here, this is shown in kind of the red color. You can see that the methylations are occurring on lysines and arginines and different residues on many different histone tails. A third type of histone modification is histone phosphorylation. So this is adding a phosphate group instead of an acetyl group or a methyl group. And this is happening on threonines and serines. And the last type of modification I will mention to histones is ubiquitination, which this is a little bit different. It's not adding just a chemical group, it's adding ubiquitin, which itself is a protein. It's a 76 amino acid protein, and it is also added to lysine residues on the histone tails. And as I mentioned before, the only thing I really want you to take away from this is that these modifications are changing the level of compaction of DNA. And here are a couple of specific examples that I'd like to illustrate. So once again, we have this diagram, which goes from our mitotic chromosome to the compact chromatin to beads on a string. What you can see here is that these beads are more tightly wound up together than these beads over here, right? Well, this is what we would call heterochromatin. Heterochromatin is usually associated with transcriptional silencing because you can imagine If my arrow is a big enzyme that's trying to get in to transcribe this gene, it physically can't access this region because the DNA is too compact. Some of the histone modifications that result in heterochromatin are listed here. This is not a complete list, but just for example, I'd like to show you how to read these. So what this is saying, I kind of find it helpful to read from the end, is it's saying that this modification is a trimethylation on lysine 9 of histone 3. This modification is a trimethylation of lysine 27 on histone 3, and so on. And these are a couple of examples of histone modifications that tend to make DNA more compact. Another couple of examples of histone modifications that instead result in euchromatin, which is opening up the chromosome. So here you can imagine that, again, my arrow is an enzyme that's trying to access the DNA. It can do it now. It can physically access this DNA and say, trigger RNA polymerase to start transcribing a gene. A couple of the types of histone modifications that result in euchromatin shown here are the trimethylation of lysine 4 on histone 3 and others. If you're wondering why some types of modifications might change the compaction of DNA, it helps to remind yourself about the chemical nature of these amino acid residues of the protein, and also remind yourself that DNA itself is negatively charged. So one thing we saw is that arginines and lysines tend to be acetylated and methylated and modified in these histones. And what do we see about these two amino acids in particular is that they are positively charged. So what you can imagine is that if you add a chemical group to these amino acids, then you are going to get rid of that positive charge. And now you are decreasing the strength of the relationship of that protein with DNA. Again, that's not always true. Some types of modifications actually increase the compaction of DNA, but this is one example of how you could get transcriptional activation or an opening up of the structure with a chemical modification of histone. And especially for this class, with these histone modifications, we're going to be concerned with how do we find them. And so I am going to go over one example today, which is ChIP-seq. So ChIP-seq, this CH part here, oh, that was a really sloppy little sign here. Let me redo that. So the CH part here is for chromatin. Chromatin, again, is DNA packaged up with proteins. The IP stands for immunoprecipitation. This is precipitating or pulling down, I'm not going to spell this right, a DNA sequence with an antibody. That's the immuno part. And then seq, of course, is sequencing. Oh, I definitely didn't spell that right here. Precipitation. To understand how ChIP-seq works, let's draw a situation where we have our beads on a string for two different regions of the genome. So here in green are my histone proteins, my nucleosomes that are made up of several histone proteins, and white is the DNA that's bound around it. And let's say that in particular, I am interested in characterizing this one modification here, let's say a methyl group on this histone that we do not see over here on this nucleosome. So the first thing that I can do with ChIP-seq is I can fragment my DNA. So let's just pull those into different fragments. And then I am going to do the immunoprecipitation part of this fun thing. So immunoprecipitation means I am going to use an antibody that specifically recognizes that methylation that I am looking for, that particular histone modification. And I'm going to pull it down because it's going to be tagged with something I can use to pull it down. And notice that that antibody that I have made here that is going to recognize the specific modification, it's not going to bind to this region of DNA over here because it doesn't have that modification, right? So now when I do my pull down, or the precipitation part that I'm not going to try to spell again, I am going to recover this bit of DNA that has that modification, because my antibody has bound that. But this bit over here is just going to go in the trash, right? Because I'm not going to pull it down. So now when I get rid of all the proteins and I wind up with just DNA, I'm only going to get the DNA that was originally bound to that histone that had that modification. So for example, I'm going to just highlight that bit of DNA in red here. So that is the only bit of DNA that I'm going to get. And now when I do my sequencing, so we've done the chromatin immunoprecipitation part, I do my sequencing and I map what I get to my reference genome, then I am going to see that this bit of, DNA that I got from my immunoprecipitation or my pull down is going to map to the genome. So I can see regions of the genome where I did see the histone modification, right? Because the only reason I was able to sequence it is because I was able to pull it down with my antibody tag, but I'm not going to see anything else because remember, everything else went in the trash. So when I get my sequences back, I map it to my reference genome and I say, this region of DNA had this specific histone modification. And now I want to switch gears a little bit and talk about the second type of epigenetic modification, and that is DNA methylation. And now we are no longer talking about a modification to a protein bound to DNA, but modification to a base of DNA itself. So here I have my naked DNA, and now I'm talking about methylation to DNA itself. DNA methylation happens specifically to cytosine residues, and specifically to cytosine residues that are part of a CG dinucleotide, where you'll often see these referred to as CpG, where the P is referring to the phosphate that connects the C to the G along this phosphate backbone of the double-stranded DNA. So that methylation occurs to that cytosine. And after that cytosine methylation happens, the methyl group is added to this fifth carbon of the ring structure, so it is now called 5-methylcytosine. And again, in this class, we are going to be concerned with how do we find these regions of DNA methylation, and the one mechanism that I will propose in this video is bisulfate sequencing. Here I'm showing you a strand of DNA, a section of DNA sequence in which we have one CG dinucleotide where the cytosine is methylated. But notice that we have another CG dinucleotide over here where the cytosine is not methylated. Now we're going to add sodium bisulfite to undergo bisulfite conversion. Now what's going to happen with bisulfite conversion is that the cytosines that are methylated will remain the same, but cytosines that are not methylated will be converted to uracil. Now remember, uracil is a base that's usually only seen in RNA. So now we're working with DNA, so we do our PCR amplification, and when we do this amplification, we are going to recover the DNA bases that you would expect, which means that that uracil is turned into a thymine at that location. And now, if we were to sequence this PCR product and map it to our reference genome, we would see that this CG dinucleotide remains intact, but this CG dinucleotide was converted in our sequence. The conclusion that we could make from this is that this cytosine was methylated and this cytosine was not. Hopefully that makes at least a little bit of sense. In class, we're going to go over some real examples of how researchers use these technologies to figure out ways that DNA and chromatin have been modified.